This method can help answer key questions in the clinical diagnostics and genetics fields, such as detecting genetic markers associated with disease outcomes, including the human papillomavirus. The main advantage to this technique is that it's a cost-effective, targeted, and time-efficient diagnostic method.
To begin this procedure, reconstitute any ordered lyophilized primers with molecular-grade water. Pull the forward and reverse primers for each multiplexed assay into a stock primer mix, such that it contains each primer at a concentration of 0.5 millimolar. After this, prepare a master mix for the PCR as outlined in the text protocol. Add 4 microliters of this mix to each well of a 384 well PCR plate, along with 1 microliter of genomic DNA at a concentration between 5 and 10 nanograms per microliter. Cover the plate with a plate cover and centrifuge for 200 times G for 1 minute at room temperature. Then run the PCR as outlined in the text protocol. Load 1 microliter of PCR product with 3 microliters of loading buffer on a 2% agarose gel. Run at 100 volts for 45 minutes to ensure that the DNA amplification was successful. Next, prepare an SAP master mix as outlined in the text protocol. Add 2 microliters of the freshly made SAP master mix to each well of the previously run.